Welcome back to Simply Sports Science, the home of clear, simple and powerful explanations for everything you need to be successful in IB Sports Science. Whether it's anatomy, physiology, training or exam revision tips, we'll break down the tricky stuff so you can learn it, remember it and use it. If you want to boost your understanding and your grades in sports science, you're in the right place. Let's take some time today to properly understand the sliding filament theory. This is a really important topic in sports science because it explains the actual mechanism of how muscles contract to produce force and movement. It can seem quite scientific at first, but once you break it down step by step, it actually makes a lot of sense. So let's start with the structure. Inside each muscle fibre, we find repeating units called sarcomeres. You can think of sarcomeres as the building blocks of muscle contraction. Line up thousands of sarcomeres end to end and you get a myofibril. Put lots of myofibrils together and you get a muscle fibre. So the sarcomere is the smallest functional unit and it's where the real action takes place. Now within the sarcomere there are two key protein filaments, actin, which is thin, and myosin, which is thick. The actin filaments anchor at the edges of the sarcomere at structures we call Z-lines, whilst the myosin sits in the middle. Imagine two thin ropes running into a thick rope in the centre. That's essentially how it's laid out. On the myosin we've got these little heads that stick out, and these are sometimes called cross bridges. They're the crucial players in contraction because they reach out, attach to the actin and then pull it inwards. If you like analogies, picture oars on a rowing boat dipping into the water and pulling. That's exactly what the myosin heads do. They grab the actin, pull it, let it go, and then repeat. But here's the key question. How do we control whether these myosin heads actually grab hold of the actin or not? At rest, actin has binding sites that are blocked by a protein called tropomyosin. It's like a safety cover, stopping the myosin heads from attaching when the muscle isn't meant to contract. Sitting on top of the tropomyosin is another protein, troponin, and this acts as the switch. So when your brain sends a signal down the motor neuron to the muscle, it triggers the release of calcium ions from storage areas inside the muscle fibre. The calcium binds to troponin, and that causes tropomyosin to shift out of the way. Once those binding sites are exposed, the myosin heads can attach to the actin. This is where ATP, the energy currency in the body, comes in. Each myosin head has an ATP molecule attached. When the ATP is broken down into ADP and phosphate, energy is released, and the myosin head uses that energy to pivot, pulling the actin filament towards the centre of the sarcomere. This action is called the power stroke. After the pull, a new ATP molecule attaches to the myosin head, causing it to release from the actin. The ATP is then broken down again, which recocks or re-energizes the head, ready for another pull. This cycle of attach, pull, detach, resets, repeats over and over, as long as calcium is present and ATP is available. Multiply that by thousands of myosin heads working at the same time and you've got a powerful shortening of the sarcomere and therefore the whole muscle fibre. Now a really important detail to remember, the filaments themselves do not change lengths. The actin and myosin filaments simply slide past each other. That's why we call it the sliding filament theory. The overall sarcomere shortens, the Z-line moves closer together, and that's what produces contraction. Let's connect this to something more practical. Imagine doing a bicep curl. As you lift the weight upwards, the brain is firing signals. Calcium is flooding into the muscle fibres. Myosin heads are pulling actin inwards and the sarcomeres are shortening. When you lower the weight back down, calcium is pumped back into storage. Tropomyosin covers the actin bind sites again and the muscle relaxes. This process is happening constantly in every movement you make, from sprinting, kicking a ball, to simply sitting up straighter in your chair. It's an incredibly efficient system and it shows just how dependent muscle function is on both calcium and ATP. Without calcium, the switch never flips and contraction can't start. And without ATP, the myosin heads can't release. And this is what actually causes rigor mortis after death, when muscles become stiff because ATP is no longer being produced. So let's recap the sliding filament theory in 6 clear steps. A nerve impulse triggers calcium release inside the muscle fibre. Calcium binds to troponin, moving tropomyosin and exposing actin binding sites. Myosin heads then attach to actin. ATP is broken down, powering the power stroke that pulls actin towards the centre. A new ATP attaches, causing the myosin heads to release and reset. And this cycle repeats, causing actin and myosin to slide past each other and the sarcomere to shorten. And that's the sliding filament theory, a cycle of chemical signals, protein interactions and energy release, all working in perfect coordination. Once you picture those rowing oars pulling actin past myosin, with calcium as the signal and ATP as the fuel, you've got the sliding filament theory nailed. Thanks for watching Simply Sports Science. If you found this helpful, make sure you like the video, subscribe to the channel and hit the bell so you don't miss any of our IB Sports Science content. We've got revision guides, exam tips and clear explanations to help you be successful. And don't forget to visit us at www.simplysportscience.co.uk. Join us again next time where we'll keep making sports science simple.